have a breaking news update straight from the University of California in Berkeley. Imagine a world where HIV can be cured and the risk of cancer is dramatically decreased. Thanks to the hard work of University of California Berkeley biochemist Jennifer Doudna, this may soon become a universal reality. Jennifer is one of the principal scientists leading the development of an innovative technology known as gene editing, specifically the CRISPR/Cas9 method. The Encyclopedia Britannica defines gene editing as the ability to make highly specific changes in the DNA sequence of a living organism, essentially customizing its genetic makeup. Scientists can now quickly destroy or edit a gene using the CRISPR/Cas9 system. According to Issues in Science and Technology, this emerging technology via the CRISPR/Cas9 system shows many signs of being cheaper, faster, more accurate, and more widely applicable than older rDNA techniques because of its ability to cut and alter the DNA of any species with ease and precision. In case you were wondering, CRISPR refers to the clustered, regularly interspaced short palindromic repeats found in most bacterial genomes. Although we have covered the definition of gene editing, let's take a look at a video created by the editor of this technology, Jennifer Doudna, explaining just how this emerging technology works.
CRISPR is a technology for changing the sequence of DNA in cells in a precise fashion to correct mutations that might otherwise cause disease. It's going to enable a lot of science to be done that was impossible to do in the past. So the way the CRISPR technology works is by the action of a protein called Cas9 that functions like a molecular scalpel for DNA. The CRISPR-Cas9 system has an amazing ability to recognize a particular DNA sequence in a cell that may be malfunctioning and then disable it by cutting the DNA. We call this gene editing, and we can use it to disable or repair a mutated part of the gene which may be causing disease.
To recap some of the information covered in the video, the CRISPR/Cas9 molecular tool is a hybrid of protein and ribonucleic acid that works by cutting the DNA at a specific sequence. The cell then takes over by repairing its DNA via two ways: gluing the two pieces back together or copying a nearby piece of DNA to fill in the missing sequence. Scientist Amber Dance emphasizes that by providing their own DNA template, scientists can induce the cell to fill in any desired sequence, from a small mutation to a whole new gene. A few of the main goals of this new gene editing technology include creating new laboratory animals for research and experimenting with eliminating disease mutations in human genes. This invention also presents an opportunity for growth in the field of agriculture, specifically in offering a new way of genetically modifying organisms that may not have as many harmful side effects as the current GMO technology. In addition to these goals, scientists even dream that the CRISPR/Cas9 system may one day alter entire populations of wild organisms. For instance, mosquitoes' DNA could be altered to make them malaria resistant, thus eradicating the threat of malaria altogether. Overall, gene editing through the implementation of the new CRISPR/Cas9 molecular tool has numerous potential benefits, including the editing of DNA to reduce the risk of cancer. Scientist Amber Dance affirms that CRISPR has the potential to improve lives, whether by altering foodstuffs to improve nutrition or yield, or by eliminating genetic or bug-borne disease. Although this emerging technology is quite different than the usual voice recognition or electric cars, I hope that by watching this video you got a better understanding of what gene editing is and precisely how the life-changing technology of the CRISPR/Cas9 system works.